unlocking the genetic secrets of hibernation for metabolic disorders. Imagine being able to hibernate like a bear, waking up refreshed and free from the clutches of metabolic disorders such as type 2 diabetes. That's the tantalizing prospect emerging from groundbreaking new research that has pinpointed specific DNA regions controlling hibernation. But here's where it gets really interesting. These findings could unlock novel treatments for some of humanity's most pressing health issues. When animals like bears emerge from hibernation, they reverse harmful physiological changes akin to those seen in type 2 diabetes, muscle wasting, Alzheimer's disease, and stroke. Scientists are hopeful that by understanding and potentially mimicking these genetic mechanisms, they could develop effective therapies for these potentially life-threatening conditions. As Elliot Ferris, a bioinformatician at the University of Utah Health, puts it, if we could tweak our genes a bit more like hibernators, perhaps we could conquer type 2 diabetes in the same way hibernators return to a normal metabolic state upon awakening. Ferris is a co-author of 2 recent studies that have identified DNA regions near a gene cluster known as the fat mass and obesity FTO locus as critical for hibernation. Interestingly, while the FTO locus exists in humans too, it functions differently in hibernating animals, potentially offering unique advantages. As Chris Gregg, a neurobiology professor at the University of Utah Health and senior author of the study, notes, what's remarkable about this FTO region is that it is the strongest genetic risk factor for human obesity. According to the World Health Organization, obesity affects 1 in every 8 people globally, posing significant risks for type 2 diabetes, heart disease, and other health complications. This underscores the critical need for effective prevention and treatment strategies. Humans already possess the genetic blueprint, explains Susan Steinwand, a research scientist at the University of Utah and co-author of the studies. We simply need to discover the genetic switches that govern hibernator traits. To pinpoint the hibernation-specific regions in the genome, the research team employed various whole genome comparison techniques, contrasting mammals that hibernate with those that do not. Ferris elaborates, if a DNA region remains largely unchanged across species over millions of years, but then undergoes rapid and dramatic modifications in hibernating mammals, it likely plays a pivotal role in hibernation. The hibernation-specific DNA regions, located near the FTO locus, consist of cis-regulatory elements, CREs, rather than actual genes. These CREs act like conductors, orchestrating the activity of neighboring genes, much like a film director coordinating different departments. The researchers discovered that these CREs regulate metabolic processes among other functions. By introducing mutations into these regions in mice, the scientists observed varied effects on weight and metabolism. Some mutations accelerated weight gain, while others decelerated it. Additionally, certain mutations impacted the body's capacity to restore its temperature post-hibernation. These findings suggest that these regions enable animals to accumulate weight before hibernation and gradually utilize their fat reserves during the cold months. Mutating a single hibernation-linked region can have far-reaching consequences beyond the FTO locus, Steinwand emphasizes. Knocking out one of these elements, a seemingly insignificant DNA segment, can alter the activity of hundreds of genes. The studies imply that CREs might also influence human metabolism. Understanding this adaptability could pave the way for improved treatments for conditions like type 2 diabetes. Moreover, these findings guide future research efforts by highlighting which DNA elements warrant further investigation. Gregg suggests there's a chance by deciphering these hibernation-linked mechanisms in the genome to uncover strategies for intervention and address age-related diseases. If these mechanisms are embedded within our existing genome, learning from hibernators could enhance our overall health. This research, published in the journal Science, opens up exciting possibilities for harnessing the power of nature to combat human health challenges. Hibernation Secrets: How Gene Regulators Control Metabolism. Neuroprotection. Imagine a world where animals can slow down their bodies to a near halt, conserving energy and protecting their brains from damage. These are the incredible superpowers of hibernators. But here's where it gets controversial. Scientists are discovering that it's not the genes themselves that grant these abilities, but rather how those genes are regulated. This revelation could unlock new treatments for human metabolic and neurodegenerative disorders. Hibernators have all kinds of secrets and biomedical superpowers that we want to understand, says Christopher Gregg, a molecular neurobiologist at the University of Utah. Hibernating animals like bears and ground squirrels can regulate their metabolism seasonally and safeguard their nervous systems during long periods of low activity. Researchers are eager to uncover these mechanisms, hoping to find commonalities that could benefit humans. In a recent study, Gregg and his team delved into the metabolic changes associated with hibernation. They found that these changes affect thousands of genes shared between hibernators and non-hibernating animals, known as homeotherms. The breakthrough was realizing that it's the regulation of these genes, not the genes themselves, that gives hibernators their unique metabolic control. Their findings, published in Science, open up new pathways for research and potential human applications. The comprehensive bioinformatics analysis in this study was groundbreaking, notes Elena Gracheva, a neurophysiologist at Yale University who wasn't involved in the research. It pushes the boundaries of what we know about hibernation and opens a new avenue for exploration. Gregg's team chose mice as their model organism because, while these animals are homeotherms, they can enter short periods of torpor, a state similar to hibernation. To identify genes linked to hibernation, the researchers used various sequencing methods to examine the hypothalamus, a brain region that controls metabolism and has been associated with torpor in mice. They observed significant changes in gene expression levels and chromatin accessibility, suggesting that a conserved gene regulatory mechanism, rather than specific genes, likely controls hibernation responses. For years, researchers in the hibernation field have sought a gene unique to hibernating animals that acts as a switch. However, Mac Yang, a biomedical engineer at the University of Southern California, believes this approach is unlikely. Instead, he suggests a central regulatory system is more plausible. The researchers also investigated conserved regions of deletion and accelerated evolution, ARs, in the genomes of hibernators and homeotherms. ARs are areas with more DNA sequence differences than expected by chance. They found about 25,000 regions of deletion in hibernators, nearly 3 times the approximately 9,000 ARs in homeotherms. This implies that hibernation may have evolved through the loss of genomic features conserved in homeotherms. This study suggests that homeotherms have genomic breaks, limiting their adaptive responses to stress, while hibernators may have deleted some of these elements, allowing for more profound and flexible biological adaptations, Gregg explains. Next, Gregg's team identified genomic structures contacted by regulatory elements that were differentially regulated in hibernators and homeotherms. They knocked out these regions and found that the knockout mice exhibited torpor-like metabolic responses, unlike their unaltered counterparts. These findings were published in a separate paper in the same issue of Science. We were thrilled to see that these individual regulatory elements seem to be functionally affecting the traits we predicted, Gregg says. Yang agrees, noting the robustness of the research. They started with big data discovery and then used mouse models to prove it, making everything very solid. Looking ahead, Gregg aims to apply his team's findings to human disorders, including insulin resistance and brain damage. If we can understand how to change the regulation of these genes, which humans also have, we might be able to develop medicines that mimic hibernation-like responses, he says. And this is the part most people miss: the potential of gene regulation to unlock new treatments for human health. By studying hibernators, we might just uncover the keys to extending human metabolic and neurological resilience, unlocking human hibernation. Key genes for obesity and diabetes. Imagine waking up tomorrow and learning that the cure for obesity, diabetes, and maybe even aging itself has been hiding inside your DNA all along. written in the same genetic code words bears use to snooze through winter. Sounds like science fiction, right? But two brand new papers in the journal Science say it's closer to reality than we ever dared to hope. Here's the jaw-dropper. Scientists have discovered that clusters of human genes are virtual carbon copies of the ones mice, squirrels, and other hibernators flip on when they power down for months without food or water. But here's where it gets controversial. Some experts argue that if Mother Nature already gave us the hardware, deliberately turning ourselves into part-time hibernators could blur the line between therapy and enhancement. Should we really be borrowing tricks from animals that evolved to survive famine? Or are we opening a Pandora's box of unintended consequences? Let's unpack what the teams actually found and why it could rewrite medicine's playbook. First, the researchers hunted for DNA regions that have stayed almost identical across mammals for more than 100 million years. Think of these as genetic paragraphs, so important that evolution refused to edit a single letter, until that is, hibernating species came along. In bears, ground squirrels, and dwarf lemurs, those ultra-conserved paragraphs suddenly sprouted rapid, dramatic changes. To the scientists, that was like spotting neon arrows pointing straight at the control switches for hibernation. Next, they looked at what those switches actually do. Instead of acting like simple on-off buttons, the genes behave more like master conductors in an orchestra. When the conductor raises the baton, hundreds of downstream genes change tempo. Some tell the body to pile on fat before winter. Others throttle metabolism down to a whisper. And still others protect nerves and muscles from wasting away. In humans, these same conductor genes are parked right next to the DNA neighborhoods linked to obesity and diabetes risk. Susan Steinwand, a genetics professor at the University of Utah and co-author on both studies, put it bluntly. Humans already have the genetic framework. We just need to find the dimmer switches. And this is the part most people miss. The real surprise isn't that hibernators have extra genetic gadgets we lack. It's that we might be the ones carrying extra brakes. Bears and mice appear to have ditched a metabolic governor that keeps human bodies burning energy at a steady, often wasteful rate. Remove or bypass that governor, the theory goes, and we could dial our metabolism up or down at will. Torch excess fat during a long weekend mini hibernation, or slow aging by reducing the cellular wear and tear that accumulates while we sleep. Picture a future where doctors prescribe a short, safe metabolic nap to reverse pre-diabetes, or where astronauts use brief hibernation cycles to keep muscles and bones strong on the way to Mars. Even age-related diseases, Alzheimer's, sarcopenia, heart failure, might be slowed if we learn to flip these ancient switches. Of course, bold ideas invite bold pushback. Critics warn that tinkering with metabolic brakes could backfire, triggering unknown side effects or creating unfair advantages for those who can afford genetic tune-ups. After all, if we can mimic a bear's ability to gain and shed weight harmlessly, do we risk normalizing extreme body weight cycling? And who decides where therapy ends and cosmetic biohacking begins? So here's the million-dollar question for you. If science offered you a safe, reversible way to hibernate for a week and wake up leaner, sharper, and biologically younger, would you take it? Or do you think we're crossing an ethical line by borrowing tricks from animals that evolved for survival, not vanity? Drop your take in the comments. We're eager to hear whether you're cheering for this genetic revolution or sounding the alarm.